Okay, good morning, students. I see only 6 of you, but we will get started. Okay. If others join, they can join later, but I don't want to keep you guys waiting. All right. So you got logged in on time. So let's get started. So just one of you confirm whether you can see my screen. Can someone confirm?
Yes, sir. So visible.
Okay. All right. Let's proceed then. So we were discussing in the last class about imaging, right? And we said that to see something, whether with your eyes or with any supporting system, we need probes. And we talked about primary probes and secondary probes. So the primary probes are the probes that are used to excite the sample we want to see or whatever we want to see. And the secondary probes are the probes that are given out by the sample. Once the primary probe hits that sample, whatever signal is given out and goes to the detector are called the secondary probes. And we also discussed that in an optical microscope or also our own eyes, the primary probes are visible light photons and the secondary probes are also visible light photons. Okay, so that was for our eyes as well as for any optical microscopy. But for electron microscopes, because we also discussed that it is not possible to see something below the wavelength of the primary probes. That means if something is visible light is the primary probe, then visible light starts from somewhere around 400 nanometers. So it is not possible to see sizes less than 400 nanometers if we use visible light as the primary probe, right? So we want to now look at materials that are very small, maybe 10 nanometers or 5 nanometers even. So to see that, we need a primary probe that has a wavelength comparable to about 5 nanometers. All right. And what are those possible probes? Cannot be visible light, of course. As the two options we have are X-rays that has wavelengths of between 2 to 5 nanometers, or electron beams in the same range. All right. So X-rays, like I told you in the last class, are very difficult to generate and the intensity is very low. And that's why we use X-rays only for very specific, studies, like I told you, elemental analysis, because every element has a different type of X-ray. And that's why once we get the X-ray, we can always verify what are the elements that are present that are sending out the X-rays. So for imaging, that means to actually visualize and see something of that size range, we therefore prefer electrons. So for electron microscopes, be it the scanning electron microscope or the transmission electron microscope, we use electron beams as the primary probe. That means we shine a beam of electrons onto the sample in order to image it. That is the primary probe. Now, what will be the secondary probe? That is something we will have to see. Okay. Now, let's proceed to the electron microscopes now. All right, so you see here uh, two types of electron microscopes. One on the left is called the transmission electron microscope, or in short, we call it the TEM. And the other one is the scanning electron microscope, or in short, we call it the SEM, right? Now, both of them have some differences in the way the imaging is done. Now, the first part is almost similar. That means we have a source of electron. Okay. So we generate huge amount of electrons. We throw them out into the atmosphere. Of course, inside the chamber, this is a closed chamber. So these are the sources of the electrons, we call them the electron guns, right? So even your TV everywhere, it's like electrons moving out, and these are shot out by an electron gun. So it's not like a typical gun we are using, but what happens is we have a source of electron. That means a material, mostly a metal, because metal, you know, are filled with electrons because of the overlap of the valence and conduction bands. So they have a huge amount of free electrons, and the best possible material to throw out electrons are metals. All right. So how do we throw out? That means we have to give some extra energy to the electrons to come out from the control of the atom, right? We already know this. But now in a metal, the electrons are actually freely moving around inside the metal. And now what we want to do, we want to throw it out of the metal. And I mentioned this term before. Again, I'm again using this term. It is called the work function. Work function. That means work function is basically the amount of energy, extra energy that you need to put on an electron to throw it out of the material. That means the electron gets enough energy to jump out physically from the material where it is confined in. So suppose we are talking about a typical tungsten wire. Okay, we have a tungsten wire, and it is a metal, so free electrons moving around, but the electrons do not have enough energy to jump out or exceed the work function. So how do we give that extra energy? The simple way of doing it is to heat up the filament. Okay, take it to a very high temperature. Then the electrons, they acquire enough energy from basically thermal energy that takes it to a very, very excited level that it can even overshoot the work function and leave the material. Okay, so basically we have some filaments here, mostly tungsten. Okay, can be some other ones also, and then we heat them up and we have enough free electrons near the filament. Okay, that means we have throwing out free electrons in the filament. Now we have to force them down into this tube, right? So the tube will force the electrons down. So how do we do that now? Again, I think I mentioned, but again I'll tell you, electrons are charged particles, negatively charged particles. All right, so we just need a very high positive voltage to pull the electrons down. So we have, you know, some electrical field here. All right, so you see here we are talking about some lenses here. In a typical optical microscope, these lenses are made of glass. In an electron microscope, although we refer to them as lenses, they are actually electric field generators. All right. So there are electric fields there that pull these electrons down. And this, depending upon the intensity of the electrical field, that means the potential difference between the electron source, all right, we call it the cathode, and the positive terminal or the anode. So depending upon the potential difference, the velocity of the electrons will be different. So typically for an electron microscope, we use in the voltage range or the electric field range of about 1 kilo electron volt to about maybe 20 to 30 kilo electron volt. Kilo, okay, that's 10 to the power 3. That's extremely high electric field. And this high electric field can actually force the electrons down. Now the job of these lenses is to converge the beam. You understand? That means if initially the beam is about, say, 1 micrometer, then gradually as it moves down towards your sample, it is collimated. You know, collimated means brought together, made into a thinner one and a much denser one, right? So you can imagine something very thick, then all of a sudden going small, 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 the number of electron is the same. That means the density will be very, very high. So finally, using these converging lenses, now these lenses, again, I said are electrical lenses and not optical lenses. These lenses can converge the electron beam, and finally, we get a very small spot size. We call it spot size, means the spot, you can say the circular spot at which it hits the sample can be sometime somewhere around 10 nanometers or even less, 5 nanometers, 2 nanometers. Okay, so that is how we heat the sample, and that is our primary probe, right? So the primary probe hits the sample. I'll talk about how the secondary probe, what are the secondary probes later. But first understand that in a transmission electron microscope, your object, that means what you are observing, you see here, it is somewhere here. This is for a transmission electron microscope. That means the object is just inside the tube through which the electron beam passes. That means, and all things are after that. That means the detector and everything is below the object we are trying to observe. That means the electron beam has to pass through the object, and whatever electron beams pass through, those are then observed by the detector. Okay, they have a screen here where the electron beams goes and fall. All right, and you can see this screen physically with your eyes because it's already magnified now. You see, collimated, that means made smaller and smaller, and then again enlarged and falls on the screen. Okay, so we can see that on the screen here. All right, so finally this goes on enlarged, and this is finally the big screen, the illuminated screen. After a lot of you know electronic lenses, we see with our eyes whatever we want to see. So the thing is here now, that's why we call it as a transmission electron microscope, because the electrons are transmitted through the sample. All right, that means it passes through the sample, and some material will absorb electrons, some will let the electrons pass by, and all those electrons will come and strike the screen here. So depending upon the intensity of the electrons hitting the screen, because it's a phosphor screen, you understand? So if the intensity, or that means the energy of the electron hitting it is more, it will give out as a bright spot. And if the intensity is slightly lesser, the brightness will decrease. And when there are no electrons reaching, it will be totally dark. So you understand? That means as the electron beam passes through the sample, some electrons are blocked by the sample. Some electrons pass through the open space. They will be the brightest because they don't lose energy. And some electrons pass through the sample. Different parts of the sample will absorb differently. Some will totally keep all the electrons, and that part will appear dark in the image. Some parts will allow some electrons to pass through, and based on how many electrons hit the screen with what amount of energy, you will see different brightnesses. All right, and these brightness variations on the screen can be translated into an image through some image development softwares. Okay, so all these electron microscopes come with a software. That means you see everything on a computer screen. Okay, now the other case for a scanning electron microscope, where is the object? You see the object is down below here. So it is not within the column actually. So just at the base of the column. Here it is in the middle of the column. Here it is at the base of the column. Okay. So what happens here? You see the detector. This is the detector. Detector is above the sample. Here the detector was below the sample. That means whatever electrons pass through are detected. Here it is not the electrons that pass through, but the electrons that are thrown out of the sample when this primary beam of electrons hit, some other electrons are thrown out of the sample. These we call as secondary electrons. So the detector picks up the secondary electrons now. All right. So that's the difference between a TEM and an SEM. And what happens, why we call it scanning? Because I told you the spot size is very small, maybe about 10 nanometer or less. So if we want to look at a sample of maybe, 1 micrometer by 1 micrometer or even bigger, then we have to scan, right? Because the spot is very small. So it's typically like your TV screen. We do call it as a raster scanning. The TV screen, the whole electron beam is not actually covering the whole screen at one instance. Okay, it is moving, it is scanning, and is moving, and it is moving at a speed which overcomes the persistence of vision of the eyes. That means the eyes cannot perceive because it is so fast. All right, if you slow it, then we will finally see that we can see these singular spots coming one after the other. But because of high frequency, our eyes cannot adjust to it, and it appears to our eyes like it is there all over the screen. All right, so same thing happens here. We scan the sample. All right, that's why we call it as a scanning electron microscope. So we scan the sample, and during the process of scanning, we are throwing out some electrons which are called as secondary electrons, and we have a detector that picks up the secondary electrons. right? Now, actually, in a typical SEM, there are two detectors. One inclined like this to pick up these electrons coming out at a slant, and there is one here also, actually here. Uh, uh, that also, it is not shown here, it is shown as an aperture is for passing the small beam of electrons here. So just near the aperture, we actually have another detector. I'll tell you why. We have two detectors in a scanning electron microscope. So if you sometimes happen to go and try to look at a sample through a scanning electron microscope, the operator might even ask you which image you want to see. Do you want to see the secondary image, or do you want to see the backscattered image? There are two types of images in a scanning electron microscope. One is called the secondary image, and the other one is called the BSE. Excuse me. BSE stands for backscattered electrons. Okay, so they will ask you, sometimes the BSE image will be much better, and sometimes the secondary electron our secondary image will be much better depending upon which process has more electrons going back. Is it the secondary electrons or the backscattered electrons? Okay. If we have more backscattered electrons coming out from your sample, then the backscattered electron detector will give a better image. And if there are more secondary electrons coming out from the sample, then your secondary image detector will give you a better image. So ultimately, it's again the variation of intensity. So based on the intensity of those individual electrons, when they hit the detector, then we get different images. Like one with high energy will give a bright spot, low energy will give a less bright spot, no electrons will give a black spot. And that's why we always get grayscale images of the images are grayscale for both transmission electron microscope and scanning. There is no color in it because it's simply intensity variation, right? Brightness, darkness. So these brightness, darkness are translated into the form of an image, and we see it as a grayscale image in the computer screen. Okay, so that is how both these operate. Now you see here this image is of a typical scanning electron microscope. Okay, it's all the electron stages are here. This part is more on the software part. Okay, so you see the screens there. So this is the electron source here. All right, the tube, what we call as the electron tube here, that accelerates the electron onto the sample. Okay, so we have the sample somewhere placed here. All right, then the detectors pick up the signal, and you see here this one image is the typical secondary electron image. All right, and you see it more like a typical digital camera we have. If you remove the color from the image of a digital camera and put it in grayscale, whatever you see is simply like a scanning electron microscope. Okay, so you will get the 3D effect like you see here. Okay, this is on a surface, and on the second screen what you are seeing, all these peaks, this is the what we call as the EDS. I already mentioned in our previous slides. If you look here, we talk about EDS, energy dispersive X-ray spectroscopy. All right, what I was talking about, that we can determine what are the elements present. So you see this is basically the EDS, and if you look here, all these peaks that are coming are because of different elements. All right, so the element will be, the software itself generates the element. This will be carbon, this will be maybe oxygen. All right, so based on the atomic number, this goes this way, and the intensity, the peak, all right, to what intensity it moves will indicate the percentage fraction in the element. That means where your electron beam is scanning, in that part, about this much in, if I say, consider this as a fraction, if that means all these peaks, if they add up to 100, then maybe this will give you 50, this will give you 30%, this will give you maybe 15%, 10%, you understand? We call it as atomic percentage. That means it will say that your sample has about 50 atomic percent of maybe zinc, 15 say 10 atomic percent of carbon, maybe 15 atomic percent of oxygen, you get all this information here. All right. So this is only from the X-rays that are generated when the electron beam hits the sample. Okay. So this is from electrons that are emitted, and this information is from X-rays that are emitted. Okay. So hope you understand this. When we hit a sample with a beam of electrons, many things happen. All right. And how we can extract, that means a lot of signals come out. And if we can gather information from all those signals, then we can actually get a lot of idea about the sample. Now you see here, this is another type of scanning electron microscope, which we call as environmental scanning electron microscope. Okay, maybe you have to note it down. I do not put it in the heading. Sorry. It is called environmental scanning electron microscope. Now, these type of microscopes are used to image living organisms. All right. Living organisms means not in the sense that during the imaging they are not alive, but these are for biological specimens. Small insects, maybe plants, cells, different types of bacteria, maybe. All of these, if you want to observe using a scanning electron, let's go see this image here. This is a SEM image of a dust mite. Small insects that move around, flying around. We call them as dust mites. All right. So this is a dust mite. We cannot see it actually with our naked eyes. It's so small. If you very clearly do, we just sit there concentrating, we might see them move a bit, but we won't see it so clearly like in this image. Okay, this may be magnified about 1000 times, all right, or 500 or 600 times at least. So that means the actual size is about 1000 of this size. So how can you see it, right? So why should we have a separate scanning electron microscope for biological samples? Because in order to get this high energy or high velocity of the electrons inside this tube, what we do, we first evacuate it. You know what is evacuation? Means to remove from there. We say, no, when there's a flood coming or tsunami coming, we say evacuate the people. That means remove those people from that particular place. So what do we evacuate here? All the air molecules. That means we make a vacuum here, a vacuum. So why do we need a vacuum? Because if there is no vacuum, then when we try to force the electron beam down, there will be so many particles in the space, like dust particles or gas molecules. Air has so many gas inside it. All those gas molecules, obviously electrons are much smaller. They will be striking those molecules and atoms and particles present there, and they will be sent in various directions, right? Like typical diffraction, it'll go in various directions, and very few will actually go down with the right amount of energy and hit the place we want. So in order to have just the electrons moving, and that too at a very high velocity without any obstruction, we need to evacuate this column. We call it as a column. Okay, this column we have to evacuate. That means we have to use a vacuum pump to force out the air from there and create a vacuum inside. Now what will happen, you see, when I say vacuum, that means we bring the pressure to extremely low value. Say maybe if we say in torr, maybe 10 to the power minus 5 torr. And compared to the atmospheric pressure, extremely low pressure. So you think about it, a membrane or something is there on one side, you have extremely low pressure, and on the other side you have high pressure. Then what will happen? That membrane will burst. That means a typical organic sample, or a sample that was alive, the pressure is mostly normal atmospheric pressure inside the body, right? Now, all of a sudden, the exterior, we force it to a very low pressure. Then what will happen? The pressure inside the body will make the body burst open. And that's why finally, when you look at the image, you won't see the actual image, but you'll look at a more of a flattened image. You won't see the dust mite like this perfectly. It will simply be a flat two-dimensional image. You understand? And that is why we cannot, when we are imaging, biological specimens, we cannot take the pressure inside the column to very, very low values. Okay. But then again, the situation is unless we take the pressure to very low values, we cannot have that energetic beam of electrons we want, because the electrons will strike other gas molecules and go helter skelter, right? So what do we do here? In an environmental SEM, we actually divide the column into a number of stages. All right. Basically, inside the column, you think about like 1 stage, 2 stage, 3 stage, 4 stage. Then what we do? The first stage will be very, very high vacuum. That means extremely low pressure inside, so that the initial acceleration of the beam or the converging of the electron beam happens. And then the next stage, we'll have slightly higher pressure. Okay, that means the evacuation is slightly less. So the beams already accelerated, get some accelerated, they pass into another one where there will be some amount of particles present. All right, even then there will be some amount of diffraction, but still, because of the initial thrust, this will go on moving, goes into the next section which has even more pressure. All right, so gradually the beam will become thinner, of course, because of some amount of diffraction happening. But if we try to keep this whole column at the same pressure, then the electrons will not reach the base. So it will be gradual increase in the pressure, and finally when it actually comes to your sample, the pressure is almost normal atmospheric pressure. And that's why the sample does not flatten out or doesn't break, right? So this type of SEMs, because of this compromise we are making on the electron beam, that means this variation in pressure, these type of electron microscopes cannot go to very high magnifications. All right. So this type of microscope can go to a maximum of about 10,000 times magnification. Whereas the other SEM, where we have same pressure inside the whole column, it can go to about 1 to 2 million times. 1,000 times, sorry, 10,000 times and million times. You see the difference, all right? But we have no choice. So that's why environmental SEMs we cannot use for studying nanomaterials. The ones we make, they can only be done for other, because 10,000 magnification is very less. Cannot really see nanostructures, but at least this type of biological specimens you can see, right? Because we cannot put biological specimens in the other SEM because it will collapse. You guys understand, hopefully. If things are going above your antennas, please tell me. Huh? So can somebody tell me, is this okay? You're understanding what I'm trying to explain? Can somebody tell me, is it understandable till now? Someone?
Little bit, sir.
Little bit. That means a little bit is not very clear, right?
Yes,
Okay,
sir.
Sir.
okay. So like I said till now, that electrons are made to hit a target because we need the primary probes. Like I told you, if our eyes have to see, it is visible light hitting the sample and getting reflected and coming to our eyes, and we see the object, right? Same thing here, we will, instead of the visible light, we will be using electron beams. So the electron beams goes and hit the sample, and we have some other signals coming out from the sample after it is hit, and these signals we pick up and then we try to image it. That means based on the intensity of the signals or in the electrons coming out, we try to see or understand from where these electrons are emitted. Okay. Now you see here, I'll just show you. See, this is suppose an electron beam coming and striking a substrate. All right, that surface of a sample you want to photograph or look at. So I'm giving different arrows because I'm increasing the voltage. That means the electric field. That means a electron column with an electric field of 30 kilovolt will have more energy. The electrons will have more energy as compared to a 1 kilovolt or zero point 0.5 kilovolt. That is okay, you understand, right? Because the electric field, stronger the electric field, more quickly will the electron be pulled. So suppose this electron beam is hitting at 0.5. This is the damage done because the electrons will enter the sample. All right, it will enter the sample, and because of physical heating, that means friction, because of friction the electrons will finally lose their energy to zero. That means all these energetic electrons heat, they'll move here inside in various directions, and then they will lose their steam. That means all these electrons pumped in at 0.5 kilovolt, they will remain within this area of the sample. Increase the voltage, more area will be penetrated by the electrons. Okay, and this is the direction. It is very, very unpredictable. You cannot say this particular electron X will go and stop here. It may not. It may stop here. It may stop here. It may stop anyway. All right, but you can actually roughly estimate the volume inside the sample where the electrons will be. Most of the 95% of the electrons will be within this area. All right, only those going very straight might be going a little further, but otherwise all will be constrained in this area. All right, increase now to 15 kilovolts, it will be a bigger volume. 30 kilovolts, it will be even bigger volume, and it can go on that way. But normally we don't go above 30 kilovolts for electron microscopy because it's a huge amount of voltage. Okay, it's very risky actually to work at such high voltages. So now you see this is the way the electrons go and stop. Now how do you predict this figure that you see here? This is generated using softwares, okay? And the simulation process we follow is a Monte Carlo simulation. You can Google and read about it. I'll just type the name for you. It's called Monte Carlo simulation. Just a minute, huh? All these things cover up everything. Just a minute. Insert. Yeah, insert. And then all these. All right. There's the textbook. Let me insert a textbook. It is called Monte Carlo simulation. Okay. Uh, might be too much if I try to discuss this at the moment, but if you read, you'll quickly understand what exactly the name Monte Carlo relate to. All right. So this is more of a drinking joint. Okay. So how people move around and all those. So this simulation roughly gives you the idea of how the electrons will move around and lose their energy. All right. And the penetration depth, that means how far inside the sample this will penetrate. Like this one is showing 3.1 micrometer. This one is showing about 1 micrometer. This is 0.16 micrometer. This is 0.01 micrometer. 0.01 micrometer will be about 10 nanometers. All right. And this is 35 angstrom. This is angstrom, right? Angstrom is 10 to the power minus 10 meter. That is angstrom, and 10 to the power minus 9 is nanometers. Okay. That means 0.1 nanometer is 1 angstrom. Okay. This is another unit that we use. And I love this unit because the lab I worked in Sweden is called the angstrom lab. Right. That's the name of a scientist. So you see, you can go down to so low list. 35 angstrom means 3.5 nanometers. That means once the electron hits at 0.5 kilovolt, the electrons will be within about 3.5 nanometers on the sample. All right. That means whatever information is coming back will be from within the 3.5 nanometers. So if you increase, more electrons will be coming out because it is penetrating more. So if you're suppose keeping your electron microscope at 1 kilovolt and you're trying to image the sample, it might not be very clear because the number of electrons coming out will be less as compared to the number of electrons here, because this is going in at a much higher velocity. So it will throw out electrons also which are more in number. So when it increase from 1 kilovolt to 30 kilovolt, we will get a better clarity on the image. Okay, better resolution, better focus, and you can say better brightness, better contrast. All right, and that's why we gradually increase if we need to. All right. Now this volume, this is like three-dimensional. Okay, you are just seeing two-dimensional on the screen, but it is actually three-dimensional. It is happening in all direction. So this actually, if you look at roughly, this is the shape of a, what you can call it, a teardrop. A teardrop. Let me bring in another one. It will be like that of a teardrop. All right. Or if you can even say it might look like garlic, a whole garlic. You've seen a whole garlic, right? That type of a shape inside the sample, all right? And that's where all the electrons will be steaming out. All right, now this is the situation as per a Monte Carlo simulation, that this is how the electrons get embedded inside the sample when we accelerate or heat it with the accelerated beam of electron. Okay, so there's a calculation on the penetration depth. All these numbers are based on this calculation. Okay, so this is the penetration depth, and these are the calculations based on the atomic weight of the material, how much of energy is coming in, the atomic number, the density of the material, all those things, they give a calculation of the penetration depth. Okay, and this teardrop-like area or volume that we say where the electrons are most likely to be present, we call it as the interaction volume. Okay, we call it the interaction volume. So because why we call it interaction? Because that is where all the interactions between these electrons coming in and the electron inside the material happen. All interactions will happen within this volume, and that's why we call it as a interaction volume. Okay, so now what happens within that interaction volume is something we need to see. Only then we will understand how your electron microscopes work, right? So let me do one thing. I have a new slide. Let me pull this figure there. Okay, let me pull this figure here. All right, and let me try to you know let me try to explain here. Okay, so if I now pick up the pen, where is it? Okay. Let me pick up the pen. All right, so tentatively you think about this. Now I said this will look like a teardrop, something like this, suppose, where more than 95% of the electrons will be there. All right. Now what will happen? There will be some electrons will be thrown out like this in a slanted direction. Okay, in a slanted direction. If you remember when I was showing you the image, the schematic diagram of the scanning electron microscope, the detector was on the side and inclined like this. All right. So what will happen? All these electrons that are going in this slanted direction, a detector will pick up. All right. We call it as the secondary electron detector. All right. So these electrons are called secondary electrons. So if I call this as the primary electron beam, or I can write it as 1 degree. So if this is the 1 degree beam, then we call this as the secondary or 2 degree. Right? Not to the power 6, huh? 2 degrees or secondary electrons. Okay, you understood? So secondary electrons come at a slanted direction. Okay, and it hits the electron. Now there will be some electron that will actually go back vertically up like this also. They'll go up vertically in the opposite direction of the incoming electron beam. And this type of electrons which goes vertically back, we call them as the back scattered electrons, or BSE. Okay, this is the name, back scattered electron. Okay, backscattered electron. Right. So this is the BSE. Right, the backscattered electrons going vertically up. There are many more signals that will come out because of this interaction, but I'll talk about them later. But since we already talked about the elemental analysis of the X-rays, I'll just mention here that there are some X-rays that are also released. Okay, so some amount of X-rays are also released. There are many other things. I'll come to those step by step. But right now, just understand that some electrons will be thrown out of the sample. Some electrons will go in a slanted direction, which are called the secondary electrons. Some electrons will go back in the vertical direction, which are called the backscattered electrons, and some amount of X-rays will be released. Okay, and these X-rays will give us the elemental analysis, what is there, the elements, and these electrons will give us the image. All right. So the meeting will end in 5 minutes. Today we have only one class, but let's try to understand this part at least before the class ends. So like I said here, the secondary electron detector will pick up the secondary electrons, and we'll have another detector somewhere here, okay, that will pick up the backscattered electrons, and we call it as the BSE detector. All right, BSE detector. Call it the BSE detector. Okay. So the detector that picks up BSE signals is the BSE detector, and the detector that picks up secondary electrons are called the secondary electron detector. Now, what is this detector? How does it actually detect this? So what happens? These detectors, both of them, the BSE or the secondary electron detector. So what is it like actually? Let me try to explain to you. The typical detector actually consists of what we call as a scintillator. Scintillator. All right. So what does a scintillator do now? So it is basically a screen where if a very, you can say an energetic particle goes and hits, then a photon is generated. Photon means a small packet of light, right? You know this. Photon is the smallest unit of a light. All right. Similarly, for heat, it is phonon, right? So what happens in a scintillator? A photon is, that means, so suppose this is a scintillator screen I'm drawing like this. An energetic particle is coming and hitting it here. Okay, there's a physical obstruction. The particle cannot go inside. But what happens? The point where it hits, exactly here, a photon will be generated. A photon will be generated on the other side. Okay, something hitting on the other side, a photon. More energy, that's what, every time an electron hits, one photon will be generated. And the intensity of this photon, how bright it will be or how dark it will depend upon the energy of the incoming electrons or incoming particles, whatever it is, whatever energy it strikes, then the intensity, all right, which is based on its energy, we say h nu, the intensity will be dependent upon the energy. That means an electron beam striking with a very high energy will give a bright photon. An electron beam striking with a lesser amount of energy will give a darker photon. All right. You understand that? So based on the intensity, automatically the scintillator will generate a photon of an equivalent or comparable amount of energy. That means when these photons all over is translated, this is a three-dimensional one, right? I'm just trying to, so finally it is something like this, suppose. This is the screen, the scintillator screen, and it is hit by electrons all over, right? This hit by electrons all over coming from the sample, and all this heating, all this electron heat will be with a different energy. So if this one is very high, this part will become bright. This is less, this part will become dark. So accordingly, the whole screen will be a variation of different intensity of light. Okay, so those coming with low intensity will be darker, those with high intensity will be brighter, and we get an image. But you have to first think about how the sample sends out different electrons with different amount of energies. That means when you see the image here with a variation of brightness, how can we actually see the actual image, or how does it relate to the actual image? Because I showed you an FCM image before. If I go back to this FCM image here, how do you actually see? You see these parts are dark, this part is slightly brighter, this part is the brightest. That means electrons going from this part have the maximum energy when it strikes the scintillator. These dark parts will have electrons with lesser amount of energy when it hits the scintillator, and accordingly you see all these contrasts. This part will be lesser energy, this has higher energy, and accordingly we can translate all those photons into an image like this. So this image is actually just the translation of photons, nothing else. Okay, that means this is the actual thing. When we bombard it with electrons, okay, suppose we are bombarding this sample with electrons with the primary beam, then when these secondary electrons move out, these electrons will move out with certain amount of energy that will give a black scale image of like this. If I hit it somewhere in this dark part, so whatever electron is going back will appear dark. Now why is this appearing dark? Because very few electrons